Hello, AP Bio. Welcome to our video lecture for Chapter 7, Cellular Respiration and Fermentation. We're going to begin this chapter, as we always do, with a picture. And you can probably tell this one is a picture of yours truly. I think the year is 2010. This photograph shows what might be the worst experience of my entire life, which is the year I decided to run the St. Joe's 5K. I don't think we have the 5K anymore, but back in the day, St. Joe's used to host a 5K race in the fall, and one year I decided to do it for reasons which you don't need to know. But like I say, in the picture, this is right as I crossed the finish line, and I might be smiling, but this race was horrible. This is the worst. This cross-country course is like the worst course ever. I saw someone with a camera at the finish line, so I gave the doofusy thumbs up and the smile, but this was torture. This was awful. I picked this picture obviously because there's a lot of cellular respiration going on, a lot of lactic acid fermentation going on. Because again, that course is awful. So if you're on the cross country team, good for you, because that's just the worst. So this chapter being on cellular respiration, this is a big one. This is a very important concept. You did this obviously in freshman bio, and a lot of you maybe didn't fully understand what was going on. But one of the things that people have a hard time with this chapter is they tend to miss the forest for the trees. They get bogged down by memorizing the Krebs cycle and memorizing how many NADs or FADs are formed. And then they miss easy questions like, I'll have kids who know how many NADH are produced by glycolysis, but they miss a question like, what gas does respiration produce, which is carbon dioxide? Or what gas does photosynthesis produce, which is oxygen? So it's important. There are some things you need to memorize in terms of numbers and molecules, but it's also important to understand why these processes are happening. It's less important that you know how many NADHs are formed in glycolysis than you know what the purpose of NADH is and what it actually does. So there are some details to memorize, but don't fall in the trap of getting bogged down by the details. So this picture just shows this is like life in a nutshell. So light energy, plants and algae, things that have chloroplasts use light energy in photosynthesis to produce organic molecules like glucose, and they give off oxygen as a waste product. Things that have mitochondria, which is all cells except for bacteria, take in the organic molecules, the glucose with oxygen, and break it down to make ATP, and then release carbon dioxide and water as a byproduct. Of this cycle, animals, humans, all animals, do the bottom half. Plants and algae do the entire thing. Common mistake is people think the plants do the top half and animals do the bottom half, and that's not true. Plants can do the entire thing. Plants, if all the animals on Earth died, plants would largely be fine, because the CO2 that plants need, they produce in respiration. They don't need, we need plants for oxygen, but plants don't need us for CO2. They can produce it themselves. Plants have mitochondria, just like we have mitochondria. Plants have chloroplasts and mitochondria, so they can store the sun's energy as glucose. Then they can use it, the glucose, they can break it down to produce energy. We have to eat to get the glucose, then we break it down to release energy, okay? This diagram here, this is super, super simple, but every year I have kids who miss things about this diagram because they get bogged down by the details. So the formula for respiration, speaking of details, you need to have this memorized. C6H12O6 is glucose. You need 6 oxygens. You need a mitochondria. And you produce 6 carbon dioxide, 6 waters, and then you give off a lot of energy in the form of ATP. And some of the energy is wasted or is lost as heat. It's where your body heat comes from. This equation, you need to know this backwards and forwards. Now, one important concept, where does the energy come from? It's going to go to ATP, but what's the source of the energy? Well, the answer is the electrons found inside the bonds of glucose, all right? CH bonds in particular, carbon-hydrogen bonds, are very, very energetic, all right? Electrons are basically the source of energy in biology, right? If I can find a way to harness those electrons and put them to work to make ATP, that would be great, and that's what cellular respiration does. So we're going to relocate the electrons in glucose, and it has a bunch of electrons. We're going to put them to work, and then we're going to break the glucose down, actually break the literal bonds down, and the waste products will be CO2 and water. That's what's left from breaking down the glucose. We need to do a quick review, what's mostly considered chemistry review of redox reactions, oxidation and reduction. Again, you likely did this in chemistry, but there's reasons why we have to go through this again in biology. So if something is oxidized, it means it loses electrons. If something is reduced, it means it gains electrons, all right? In this equation here, sodium goes from Na to Na plus, which means the sodium, if it became charged positively, it means it lost an electron. So in this reaction, the sodium is oxidized. The chlorine goes from chlorine to Cl minus, which means it gained a negative charge, which means it gained electrons, so it was reduced. There's an old mnemonic, or not mnemonic, just an old saying. It's about the lion. Leo, the lion, goes grr, right? Leo stands for loss of electrons is oxidation, and the GER, G-E-R in this case, stands for gaining electrons is reduction. This you need to know. You need to know that oxidation means you lost electrons. Reduction means you gained electrons. If you lose electrons, you become more positively charged. If you gain electrons, you become more negatively charged. This is just the same thing, just showing what's oxidized and what's reduced. In the formula, so this isn't always the case what I'm about to say, but it's the case enough at the time where it's worth saying. So this is showing the oxidation of methane. So notice the carbon goes from being bound to hydrogens to being bound to oxygen. And if you go through a reaction where you increase your number of bonds to oxygen, you are probably oxidized. So notice the carbon goes from CH4 to CO2, it's oxidized. Here, my oxygen, which was O2, those bonds get changed to bonds that are hydrogen bonds. If I give you more hydrogens, you've probably been reduced, okay? That isn't always the case, but it's the case usually for AP biology. If you gain bonds to oxygen, you're probably oxidized. If you gain bonds to hydrogen, you're probably reduced, right? It's just a good thing to have in the back of your head. So electrons have energy, right? We've already been through this. When you move an electron to an atom with a higher electronegativity than what it had before, it loses energy, right? The most electronegative atom is fluorine, right? But biology doesn't love fluorine. Right under fluorine on the periodic table is oxygen. So if I move electrons to an atom of oxygen, they have lowered their amount of energy, all right? That's why a CH bond to a CO bond is oxidized, and you've lowered the amount of energy that those electrons had. So if you look at the equation for respiration, I'm gonna go from glucose, and eventually the electrons are gonna wind up in water, right? Oxygen gas is the final receptor of the electrons in the electron transport chain, and it goes from oxygen gas just to water. So in doing that, you've lowered the energy of those electrons, which is also kind of what happens. You know, I could just burn glucose. It would be pretty stinky. You give off some heat, great, but you haven't captured the energy if you just burn it. So we need to find a way of capturing the electrons of glucose and putting them to work. And the way that you capture electrons is you use what are called electron shuttles, which is NAD and FAD, all right? NAD, which stands for nicotinamide, and FAD, which I forget what FAD stands for, are molecules that can be alternatively oxidized and reduced. NAD plus, what's in bold here, is a coenzyme. It's a molecule that can accept electrons. If you give NAD plus electrons, it forms NADH. And if you look at the formulas, you don't need to know these structures, but look at this part of the molecule right here, and see right here it's gained another hydrogen. So this is the oxidized form, this is the reduced form. The reduced form has electrons, and it has an extra hydrogen, which is why it's NADH, all right? Let me ask it again. This is a common question. Between NAD plus and NADH, which one is the oxidized form and which one is the reduced form? Think to yourself before I say it out loud, all right? The oxidized form is NAD plus, and the reduced form is NADH. And the reduced form has the electrons, okay? FAD and FADH2 is another electron shuttle, which we'll see in a minute, which also carries electrons. FAD, FAD is the oxidized form, FADH2 is the reduced form. So once you've captured the electrons, they're going to go to a structure called the electron transport chain, which is in the mitochondria. And this is a long series of steps that we'll go through later in this video. But basically, at the end of the chain, you have oxygen, O2 gas, which sort of pulls the electrons through the chain. And as the electrons go through the chain, if you look at this diagram, this kind of shows it. It shows like little turbines being turned. As the electrons go through the chain, which is part of the inner membrane of the mitochondria, stuff happens that produces ATP. At the end of the chain, the oxygen gas accepts the electrons and forms water, which is why the process produces water molecules. Okay, now this is the stuff where kids get bogged down by the details. The AP exam doesn't often ask you lots of details about this stuff. Sometimes they might give you diagrams to interpret, or they ask you very detailed questions about a diagram, but you have a diagram. So don't get bogged down by the details, all right? The things that you need to have memorized, I will tell you as we go through it. So there's three main parts of cellular respiration. There's glycolysis, there's the Krebs cycle, and there's the electron transport chain. This just shows a preview. One thing that you definitely need to know is where each process happens. Glycolysis happens in the cytoplasm. The Krebs cycle happens in the mitochondria. And the electron transport chain also happens in the mitochondria. For one molecule of glucose, you get 30 some odd molecules of ATP. I mean, there isn't one specific number. It depends upon a couple of different variables. But if you know that you're in the 30s range, then you're fine. Your textbook uses this color coding. This teal color is glycolysis. This salmon color is the Krebs cycle, or the citric acid cycle is the same thing as the Krebs cycle. And this kind of purple color is the electron transport chain. And this just shows the whole thing. Glycolysis, here's a big mitochondria, pyruvate oxidation, we'll get to in a minute, the Krebs cycle, and then the electron transport chain. When we're done with this lecture, this picture should make complete sense. So, okay, let's begin with glycolysis. So the word glycolysis means sugar splitting. Glyco refers to sugar, like glucose, and lysis means to split. One thing that's useful to do is to track your carbons. Remind yourself how many carbons one glucose has. It has 6, all right? Glycolysis, the end result is going to break down that glucose into 2 molecules of pyruvic acid, or pyruvate, same thing. They have 3 carbon atoms, which is why you have 2 of them. So you went from 1 glucose to 2 pyruvates. Have you lost any carbons? No, they're all still there. When you lose carbons, where do the carbons go? Well, they're lost as carbon dioxide. The CO2 that you are breathing out right now came from your mitochondria, or in this case, it came from the cytoplasm of your cells doing glycolysis. If you don't lose any carbons, does glycolysis produce CO2? It does not, okay? Actually, what I just said is incorrect. Glycolysis does not produce CO2. So the carbons that you breathe out in carbon dioxide come from your mitochondria. They do not come from your cytoplasm. I just said something incorrect. Hopefully you caught that. Glycolysis does not require oxygen, so it can happen under anaerobic conditions. Yeah, okay. So it happens in the cytoplasm. You start with 1 glucose, you end with 2 pyruvic acids. This slide is decently important. Glycolysis is a 10-step process. You don't need to know what the 10 steps are. This second point, the yield that you get, this you need to know. And the numbers, in this case, I would actually have the numbers memorized. So we already said you get 2 molecules of pyruvate, right? You also get 2 NADHs, all right? We're not going to use the NADHs yet, but it makes 2 NADHs, which we're going to keep in our back pocket for later. It also produces 2 ATPs, which is a nice benefit. You know, if it can net 2 ATPs, why not? One thing to note, it's worth saying, it actually produces 4 ATPs, but you need 2 of them to begin glycolysis. So you need 2, you make 4. Overall, you make 2. Sometimes the APICSM will give you a diagram and you'll see how you actually made 4 and kids get confused. Well, you did make 4, but you needed 2. So your net is 2. Notice you don't produce any carbon dioxide. You haven't lost any carbons, and you didn't need any oxygen. Okay. This slide just is a diagram of what I just said. You needed 2 ATP, you made 4, so you made 2 overall, 2 ATP, and 2 NADH, and 2 pyruvates. Okay. We don't need to go through this. I put these slides here just to show you what the 10 steps are. Just notice we begin with glucose. Each step requires an enzyme. So hexokinase, phosphoglucoisomerase, phosphofructokinase, aldolase, these are enzymes. You don't need to know these names. But it is worth noting that each step needs a specific enzyme. And this is the second half of the 10-step process of glycolysis. Some of these names are just fun to pronounce. Phosphoglycerokinase, you should name your first kid that. They'll have a terrible kindergarten year. Okay, so there's 3 steps, right? Well, there's also a sort of a step 1 asterisk because there's an intermediate step. So, you know, where are the pyruvates made? They're made inside the cytoplasm. Well, I need to get the pyruvates into the mitochondria to do the Krebs cycle. There's a step called the intermediate step. Some books call it the link reaction. The same thing. This is a step that happens inside the mitochondria, and it takes pyruvic acid and it turns it into acetyl-CoA. All right. Now, pyruvic acid has 3 carbons. Acetyl-CoA has 2 carbons. So where did that extra carbon go? It got lost as carbon dioxide. And because, you know, 1 glucose became 2 pyruvates, those 2 pyruvates become 2 acetyl-CoAs. For glucose, you lose 2 carbon dioxides because, again, 1 glucose gives you 2 pyruvates. All right. So at this point, there were 6 carbons. We lost 2 CO2s. There's 4 carbons left. Where are those 4 carbons? They're in the 2 acetyl-CoAs. This also produces 2 NADHs, which is, again, I'll keep them because we're going to use them for later. Okay. All right, cool. So at this point, we're inside the mitochondria. We're in this diagram. So the Krebs cycle. 1 turn of the Krebs cycle is 1 acetyl-CoA. And because a glucose gives you 2 of those, the numbers that are on this slide are per glucose, which is per 2 turns of the Krebs cycle. So the Krebs cycle, let me just say it again, turns twice for 1 glucose. We're showing it turning once because it's 1 cycle. The things that you get, this you should memorize. And again, this is per 1 glucose. You get 2 ATP, you get 2 NADHs, you get 2 fat H2s. And just to remind you, fat H2 is the reduced form. The oxidized form is just fat, just FAD. There's no plus. It's just FAD. And you get 4 CO2s, which completes our oxidation of glucose. Because remember, we lost the 2 CO2s back in the intermediate step. Every year, kids get confused because they see the diagram shows you're only making 1 ATP, but you turn the cycle twice for 1 glucose, right? Now, at some point, someone probably made you memorize the Krebs cycle, which is what's pictured here. I had to memorize it when I was in school. I had to commit it to memory when I was in college. You don't need to have this memorized for AP Bio. No one would ever have you draw it. Honestly, memorizing it, unless you're like a biochemist, there's really no need to memorize it. But you need to be able to use it, all right? Let me ask you a few questions on the Krebs cycle. The AP exam would give you a diagram of it. There was actually a 10-pointer a couple years ago where they gave you the Krebs cycle and they asked you questions about it. And that question, kids bombed, even though they had the diagram. So let's just do some practice. So notice acetyl coenzyme A, acetyl-CoA is what's coming in. If I asked you what does acetyl-CoA, okay, back up a second. When acetyl-CoA combines with oxaloacetate, what do you get? The answer is you get citrate, right? Citrate then turns into isocitrate, which turns into alpha-ketoglutarate, and so on. Why is it important that we refresh or that we regenerate oxaloacetate? Well, if you have no oxaloacetate, acetyl-CoA can't enter the Krebs cycle. So why does this have to be a cycle? Because I have to regenerate oxaloacetate to accept more acetyl coenzyme A, right? So if step 8 doesn't happen and I have lots of malate and no oxaloacetate, the cycle just ends, all right? Another question. If glycolysis stopped and if your supply of acetyl-CoA dried up, you had no more acetyl-CoA, of all these molecules, which one would increase the most? Well, the answer is oxaloacetate would increase the most. If I stop this step right here, if I put like a blockade right there, oxaloacetate builds up, right? If my supply of acetyl-CoA goes away, which molecule is the first to start to diminish? Well, citrate goes down. because oxaloacetate and citrate combine, oxaloacetate and acetyl-CoA combine to form citrate, right? If I blocked step 7, which molecule would go down? Malate would go down because it's after. Which molecule would accumulate? Fumarate would accumulate because that's right before the blockade, all right, or the barrier. Blockade sounds very dramatic. So yeah, so you don't need to memorize this, but you need to understand every step if given a diagram, all right? Okay, cool. This just goes through the diagram with each part blown up. Like when you see this, this shouldn't scare you. You should be able to use this as a tool to get the questions right. Okay, so that's all for the Krebs cycle, right? We're done with the Krebs cycle. By the way, this is dumb, but it's also called the citric acid cycle because the first thing you make is citrate, which citric acid is a form of citrate. Okay, so the electron transport chain. So the electron transport chain happens inside the mitochondria, specifically the inner membrane, that highly folded membrane that formed the cristae of the mitochondria. So what have I accomplished so far? So you've made, I guess 4 ATP. You made some ATP, that's great. 4 is not a whole bunch. You've made some carbon dioxide, which isn't, we just breathe it out. It's a waste product. But you've made a lot of NADH and FADH too. So you have lots of high energy electrons stored. So let's do something with them. The term oxidative phosphorylation, which is what's on this slide, this is the term for using the NAD and the FAD in the electron transport chain to produce ATP, all right? The last 2 points of this slide are on the picture that I'm about to show you. Actually, let me show you the picture and then come back to this. So, well, okay, at this point, we're in the purple. We're in the electron transport chain. So this shows the electron transport chain. So this is the inner membrane of the mitochondria. Now, that membrane is highly folded. What does highly folding a membrane do to it? It increases the surface area. So the more surface area you have, the more of this that's going on. Remember my analogy with the more expensive towels, and the more towel, the more fabric, the quicker it dries you off because it absorbs more water. Cheap towels are very thin and have the threads are bare and thin. So these molecules, FMN, FES, all of these things, these are called cytochromes. And cytochromes are molecules that cytochromes are found. Cytochrome C is one that's very, very famous because it's found in almost every kind of life on Earth. Some bacteria might not have it, but cytochrome C is a very, very, very common protein that virtually all life has. Anyway, so cytochromes are molecules that you can alternatively oxidize and reduce. So NADH comes in, look at the diagram, and it gives its electrons to this first cytochrome. Then it kicks off NAD+. So the NAD got oxidized, and this FMN, whatever that stands for, I don't know what it stands for, is reduced. Then this one's going to reduce that one. It reduces that one. It reduces that one in a chain. And when Q reduces cytochrome B, Q gets oxidized because it gave the electrons to the next one. So in this stepwise process, you're just transferring the electron from one cytochrome to another one. At the end, I got to get the electron out. If I can't get the electron out, the whole thing clogs up. And at the end, see this O2 gas? This is why you need oxygen. This is it. The O2 gas accepts the electron to form water. If there's no oxygen, the electron transport chain gets clogged, it stops, and you die. That's why you need oxygen to survive. Okay, so these last 2 points that I skipped, most of the chain's components are proteins called cytochromes that alternate between reduced and oxidized states, and the electrons drop in free energy as they go through the chain. So the question is, the free energy that gets lost, what's the cell using that energy for? We don't do this just for giggles. There's a reason why it's doing this, and the reason why you'll see in just a minute. This slide has a bunch of words, but this explains what's going on. Let me show a diagram, then I'll come back to this slide. I'm going to come back to that one. So this is a different diagram of the electron transport chain. So as the electrons go through the chain, and notice FADH puts its electrons in a little bit further down the chain, the free energy that's lost is used to pump protons from the matrix into the intermembrane space. This yellow represents the matrix, and this orange represents the intermembrane space. these protons, these hydrogen ions, are being pumped against their gradient. So this is active transport, right? What therefore happens is the protons build up in the inner membrane space, and they want to go back down their gradient. The only place where they can go back down is this protein. It's an enzyme called ATP synthase. The name gives it away. When the protons finally get released, they get relieved, their pressure, I guess they scream. When they go back down through this channel, that energy is used to turn ADP and inorganic phosphate back into ATP. And that process is called chemiosmosis, all right? So going back to all these words, so as the ETC is capturing the electrons, as they get pumped from cytochrome to cytochrome, they pump protons from the matrix to the inner membrane space. And that creates a concentration gradient, which we call a proton motive force. Protons, because these are just protons. Motive, because it's like motion. They want to move back into the matrix. The question is, how do I therefore use the proton motive force? Well, I use it in ATP synthase, because when they come back into the matrix, they go through ATP synthase, which that energy is used to make ATP. The term chemiosmosis, there's lots of fancy words in this chapter. Chemiosmosis is the term that we use. The term is when you use a proton motive force to make ATP, that's called chemiosmosis, using the proton gradient to do some kind of work, which in this case is to make ATP, all right? So the whole process is called the electron transport chain. The electrons create a proton motive force, which is used to make ATP in a process called chemiosmosis, all right? This just shows a diagram of ATP synthase. It's actually really cool. It's kind of like a rotor. The little thing spins, and you put ADP and PI in it, and when it spins, it puts them together into ATP, and it spins when the protons go through it. So it's like it has a physical like rotary system. It's really pretty elegant actually. Now, the term oxidative phosphorylation. So this is a really good diagram. So the electron transport chain created the proton motive force. Chemiosmosis uses the force to make ATP. The whole thing is called oxidative phosphorylation, all right? Back in glycolysis and in the Krebs cycle, we made a couple ATP, right? Glycolysis made 2, the Krebs cycle made 2. Was that through an electron transport chain? No, it wasn't. That was just an enzyme that made ATP, right? That's called substrate level phosphorylation. I should have mentioned that back when we did glycolysis. Making ATP just using an enzyme, you know, enzymes have substrates, right? That's glycolysis, that's the Krebs cycle. That's called substrate level phosphorylation because it just uses an enzyme, all right? Excuse me. If you make ATP using an electron transport chain, that's called oxidative phosphorylation, okay? Sample AP Bio multiple choice question. Which of the following terms does not belong with the others? And there's 4 choices. The terms are electron transport chain. Actually, you know what? I'm going to give you 5 terms. Electron transport chain, B is chemiosmosis, C is oxidative phosphorylation, D is proton motive force, and E is substrate level phosphorylation. Which one doesn't belong? E doesn't belong because that's what happens in glycolysis. So, electron transport chain, chemiosmosis, oxidative phosphorylation, proton motive force, those are all talking about the same thing. Substrate level phosphorylation is the earlier process, okay? All right, this slide's a summary, all right? This little flow chart that I have right here is kind of useful. It shows where the energy goes. Energy begins in glucose, then it goes to NAD and FAD, then goes to the electron transport chain to create the proton motive force, which then goes to ATP. Fun statistic, only about 34% of the energy of a glucose molecule actually goes to make ATP. The rest is lost as heat. So it's not a very efficient process. It's very wasteful. Of course, the heat, that's where you get body heat from. So we do use it, but it's not used for ATP. 30 to 32 moles of ATP, it's in the 30s. Why the numbers are different, we don't need to worry about it. It's in the 30s. All right, and this slide reviews everything, right? At this point, this diagram should make perfect sense. If it doesn't, go back and look at the processes again and see how they all fit together. Okay, so we're going to end this chapter with a word on what if there's no oxygen, right? What is anaerobic respiration? So, aerobic means you use oxygen. Anaerobic means you don't use oxygen. One thing I want to point out, so glycolysis earlier in the video, pretty much every cell on Earth does some form of glycolysis. You do it, plants do it, roaches do it, algae do it, fungi do it, bacteria do it. Glycolysis is almost like a universal process. Remember, glycolysis produces 2 ATP, which is all that bacteria can get from sugars. And for bacteria, that's fine. You get 30 some odd, so you're way more efficient than a bacteria, but you also need more energy than bacteria do. If there's no oxygen, you do a process, or bacteria do a process called fermentation. And fermentation is basically just doing glycolysis, doesn't need oxygen. But remember, glycolysis made pyruvic acid and made some NADH. I have to get rid of that stuff if I want to do more glycolysis. So fermentation adds an extra step to regenerate the NAD plus and to get rid of the pyruvic acid, all right? So the purpose of fermentation is not just to make the 2-glucose, it's also to regenerate NAD plus and to get rid of the pyruvate. Again, using fermentation to make ATP would be substrate level phosphorylation because there's no electron transport chain. The 2 types, alcoholic fermentation and lactic acid fermentation, are on this slide. So let me just also make the point. If I have no way to regenerate the NAD plus to get it back, then glycolysis stops. If all my NAD plus becomes NADH, that means you did lots of glycolysis. That's great, but if I have no more NAD plus, I can't do glycolysis anymore. So getting the NAD plus back is another thing you need to do in fermentation. So alcoholic fermentation, the pyruvate gets turned into ethanol. This is how we make alcohol. Basically, you're using yeast to do the fermentation. What you feed the yeast, whether it's barley for beer or grapes for wine, it depends as to what you want to make. Lactic acid fermentation is what happens in your muscle cells. Muscle cells in a pinch can do fermentation. If oxygen is very scarce, say you're exercising and your oxygen levels get low, your muscles can, for a short period of time, do fermentation. The byproduct there is lactic acid, which kind of burns in your muscles. To get rid of that, you need to breathe. You need more oxygen, because oxygen, if I combine lactic acid with oxygen, it'll break back down into pyruvate or whatever it breaks down to, which causes the muscle pain to go away. The only cells in your body that can do fermentation are muscle cells, which kind of makes sense because, muscle cells would be the first to be starved for oxygen because they use the most oxygen if you're under stress, like physical stress. This picture just shows turning glycolysis into pyruvate. That's glycolysis, turning glucose into pyruvate, sorry. Alcoholic fermentation turns it back into ethanol, and lactic acid turns it into lactate or lactic acid. Okay. Doing fermentation, which again is anaerobic, you only get 2 ATP per glucose versus the 30 some odd in respiration. That's better than nothing. Things that are obligate anaerobes, like if you're obliged to do something, you have to do it. If you're an obligate anaerobe, it means you cannot have oxygen. Oxygen is actually a poison. Some kinds of bacteria are poisoned by oxygen. They would have to live in places like the bottom of the ocean or the bottom of a salt marsh or somewhere where there's no oxygen. Pretty exotic places. Facultative anaerobe can do either or. So like yeast can do respiration or fermentation, depends upon if there's oxygen. Your muscle cells are facultative anaerobes because they can do either one. Every other cell in your body this word isn't on the site, but if you back up, if you're an obligate aerobe, that means you have to have oxygen. So all the cells in your body are obligate aerobes except for muscle cells, which are facultative anaerobes. This just reviews glucose through glycolysis forms pyruvate. Either goes in the mitochondria to do respiration, or you do fermentation to make ethanol or lactic acid or whatever other byproduct that the cell does. Last thing. So I mentioned that pretty much all cells on Earth do some form of glycolysis. And glycolysis does not produce or does not require oxygen, right? So looking at the fossil record, there are cells about 2.7 billion years ago that were doing glycolysis. And going that far back, Earth's early atmosphere had very little oxygen. Today it's about, I think, 16% oxygen. until you have plants and algae, Earth's oxygen is very, very scarce. The oxygen that we have comes from plants and algae. And those first cells would be little bacterial things that are not doing photosynthesis. So if cells going back 2.7 billion years were doing glycolysis, and all life today does glycolysis, that tells you that that process is a very, 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 very ancient process. This is an important theme that's going to come back several times in AP Bio, where if you want to learn like how old something is, a process, see how many things do it. All right, if everything does it, it probably evolved very, very early in the history of life. Let me give you an example. A molecule like hemoglobin, you know, you have hemoglobin. It's common in animals. Roaches don't have hemoglobin. They have a different kind of respiratory pigment carrying oxygen. So hemoglobin is a newer thing than glycolysis because fewer things do it. Does that make sense? The more things that do it, the older a process it must be. If everything does it, that means the first cell probably did it, which means the process is a very, very, very ancient process. Okay, that's enough for one video. I hope that was helpful. I will see you guys next time.